The title of Alan's talk will be The Limits of Epidemiology for Food Post-Market Surveillance Lessons from the Olestra Post-Marketing Surveillance Study.
Well, thank you for inviting me here. It's a pleasure to talk to this group. I should say at the outset that I inhabit a different world than most of you. I have no laboratory. Is there a mic? Yes. Oh, yeah, there's a mic. I inhabit a different world than most of you. I don't have a laboratory, and it's interesting how, and we'll talk about this a little bit later. It's interesting how there's some similarities between the types of science we do that I'll bring up later that I just found very interesting and quite unusual. So I'm going to talk about Olestra post-marketing surveillance, which is something that my group was asked to do well over 10 years ago. And just a bit about...
The other one.
Oh, just a bit about what Olestra is. So Olestra is a fat mimetic, and it's a fascinating substance in that it can be modified by changing the fatty acid composition of the fatty acid esters to take on flavors and functional characteristics and organoleptic characteristics that entirely mimic various types of fat. So you can use it to bake, you can use it to fry, you can even make spreads out of it. It was discovered in the early 1970s when scientists at Procter & Gamble were trying to come up with fats that would be more easily absorbed by infants. And so they did these experiments of creating fatty acids that, glycerol would be the triglyceride that we normally consume that had 4 or 5 and up to 8 esterified fatty acid groups. And it turns out it worked in the opposite direction. So the more esterified fatty acid groups, the less you could absorb it. And actually, when you get to about 7, it's not absorbed at all. So that's where this stuff came from. And it looks like this. And you can imagine the complexity of digesting something like this when normally we would digest something that looks like that. And this is the history, which is interesting. So in '71, Procter & Gamble first met with the US FDA and wanted to know how they could go about getting approval for this substance. And the original indication they were interested in was cholesterol reduction. And it turned out that it does reduce cholesterol, but less than 15%, and therefore the FDA was not interested in approving it for that indication. By '86, they decided to apply for approval for general marketing for all types of food. This was very broad, and it absolutely stumped everybody. FDA didn't know what to do with it. And so P&G narrowed their application to just cover savory snacks. This is in 1990. And that application hung around the FDA for 5 years. There were many committees, there was much talk, but nothing happened. No one could make any decisions. And so there was, in 1995, there was actually a congressional subcommittee meeting on the fact that the FDA could not move its work along. That is what finally promoted approval. So there was a special regulatory team set up at FDA that voted 17-5, and finally in 1996 it was approved, but based on conditional upon post-marketing surveillance. And I just take these words from the final 1997 FDA ruling because they're kind of interesting. So Procter & Gamble has made a commitment to the agency that will conduct the studies outlined in the letter to FDA, which we'll talk about in a minute. And if Procter & Gamble does not conduct the studies, FDA will consider the approval set forth in this document to be void. So if they didn't do the post-marketing surveillance and they had a 2-year timeline, it was going to be pulled from the market. And this is a sort of statement on FDA of how they see their responsibilities. It's the agency's responsibility to review and evaluate data generated by these studies, and interesting as well, to evaluate any data that bear on the safety, including related to the health significance of carotenoids. And a little bit of nutritional history for those of you who aren't part of this. 20 years ago, beta-carotene was going to, in supplement form, going to prevent cancer. And we had all kinds of very strange ideas about what carotenoids were going to do to health. And these have fallen one by one. Beta-carotene actually increased increases cardiovascular disease and cancer risk in certain populations. Lycopene turns out to do nothing. But at the time this was passed, there was great hope. We were still thinking about hope for these compounds, so there was a problem. So, and such were born these chips. And I just want to say that this to me was a marketing nightmare. First of all, the name of the chips, wow, is actually kind of offensive. And they chose to, these products actually tasted different than the standard products. And I don't know who they were after in terms of taste, but I think they went for the wrong market. I'll give you an example. These nacho cheesier, wow, corn chips tasted unbelievably chemical. And it's hard to understand why they did that. But so they did, and this stuff is released in the market. And so this is what we knew about it at the time it came to market. So this is a study that looked at feeding humans for 8 weeks. Whoops. 8, 20, and 32 grams of Olestra per day. Just to put that in context, a 1 ounce serving of potato chips, which isn't very much, is an 8 gram serving of Olestra. So a sort of a standard potato chip serving would be about 12 grams. So that puts that in context. And so what we knew was that there were striking decreases in the carotenoids, small decrease in tocopherols, small decrease in vitamin D, and a relatively substantial decrease in vitamin K. And these are all statistically significant, and so we went into it knowing this was true. And in particular, what people were excited about was the fact that a relatively small dose of Olestra, 3 grams per day, could lead to striking reductions in the most lipophilic carotenoids, which is lycopene. So you almost got a 40% reduction in lycopene from just feeding 3 grams a day. So there was concern about when this substance was released into consumers, were they actually going to be at risk of something because of reduced carotenoids. The other thing we knew is that you had to eat the high carotenoid food at the same time as you ate the Olestra-containing food to see the effect. So we call that co-consumption. And this is an interesting experiment done with pigs, and it has one of my favorite lines in it in the results. The results start, the pigs ate the chips readily. But here's the effect when you mix the chips into food, and here's the effect if you feed the chips as a snack, in other words, in between meals. So you see very little effect unless the compounds are co-consumed. And the other thing we knew is that because the fat was not absorbed, there was a laxation effect. And so very striking increase in GI symptoms. So for 18% of the days that people were eating 32 grams per day or more, they reported GI symptoms, and the most common ones are diarrhea and cramping. So we could expect this. I just want to say, parenthetically, this looks exactly like sorbitol. So it's although the graphs look striking, this is very similar to the laxation effects of other approved food additives. So this is what Procter & Gamble promised to do to the FDA. They had this letter, and they said, well, we're going to monitor Olestra intake. We're going to look at how food consumption patterns change because Olestrol is introduced, and we're going to look for changes in nutritional status. And I'll take a side tour here and mention that gastrointestinal symptoms were not evaluated in terms of post-marketing surveillance, and that's because there was a decision made to evaluate them instead in terms of randomized clinical trials. I don't want to show you these results because they're kind of fascinating. This is a study that was done by taking roughly 1,000 people, inviting them to a movie theater, and they could go to any movie they wanted to see. I think it was an Octoplex or something, so they had 8 movies. And they got an unmarked, very large bag of potato chips, and I mean large. And so they would basically eat them ad libitum, and no one finished all the chips, so we knew we gave them enough when this was done. And And this is just one acute feeding, so it's just once. And you can see there's absolutely no difference in the reporting of GI effects up to 3 days after. So that's the acute effects, at least in the amount that people eat, are relatively undetectable. And this was a study that P&G did with roughly 3,000 people, and here they gave people unmarked bags of chips and they took them home, and it was a household-based intervention. And I believe it was 6 weeks. And again, you can see there's almost no difference in all of the GI effects that they were looking at. And the other thing to note when you think about post-marketing surveillance is that a lot of the things you look at as outcomes are actually quite common. And so sometimes distinguishing between attributing a very common outcome to an exposure is extremely difficult. Because of who I knew, actually, is the way this worked. A series of P&G said, what does post-marketing surveillance mean and how do we do it? Because there was simply no model. And it somehow filtered down to me, and we said, yes, we can think of something to do. And we first decided to at least specify what the aims of post-marketing surveillance were going to be based on what we knew about the effects of Olestra in these various trials. So first we'd look at the how much Olestra is being used, who decided to use it, and what were their characteristics, and how did they use it, what were their use patterns. Then we wanted to see if there was any relationship between the introduction of these foods and serum micronutrients in the U.S. population. So that's a population question, which means we had to make the inference about the introduction of these foods into an entire group of people. And the second question was, within people actually using Olestra, is there any evidence that using Olestra has effects on serum micronutrients? And here we were looking at long-term heavy users. Both of these questions were powered to see a 10% decrease in carotenoids or fat-soluble micronutrients. And the design, so we had 3 aims and we had 3 essential designs. The first design was a random dial digit survey. And we started just by using random digit dial, using what's called a list-based method. You buy lists of phone numbers that have been pre-screened, and they know they don't go to businesses, and they know the numbers work. So that helps you a lot. By the end of this exercise, we were sending express mail letters to people saying we want to call them, and we included a $10 bill to get them to participate. So the response to RDD, random digit dial surveys, is right now quite poor. Among these people, we did a baseline survey before the introduction of Olestra foods, and then we repeated this 1, 2, and 3 years after. So there are basically 4 cross-sectional samples. For the second question about population micronutrients, people who completed the telephone survey, we invited them into a clinic. And here we offered them $100 for the individual, and if they wanted to bring a child, we gave them $200. So this is substantial bribing, and it's really necessary because, as you'll see later, it's a very onerous study protocol. So we got a group of these people in before Olestra was introduced, and again we got repeated cross-sectional samples at years 1, 2, and 3, recruited the same way. And then for the last one, looking at long-term heavy Olestra consumption, for the first clinic cross-sample group, so the ones who were recruited to come to the clinic before Olestra was introduced, we gave them calls every 3 months. And we asked them, it's a very short phone call, and we just got information about whether or not they were, eating snack chips, and if so, what type and how often. And we selected from those the people who reported using Olestra at least once. So we recruited 80% of, so we got a population that was consisted of 80% those people, 20% of people who did use Olestra. And we got them to come repeatedly to the clinic. So this is the first group, and it's a cohort, and we tried to follow them for 4 years. Again, giving them relatively large sums of money to participate. And so what you can see this look like, and the study was done in 2 waves. The Olestra chips were introduced in a test market in Indianapolis, Indiana, which happens to be where P&G is located, about almost a year before they were introduced nationally. So we had essentially a sentinel site which we could use to work out our methods and make sure this, exactly how this would go. And then we could move to a national study when they were released later. And this just shows you the numbers of people involved. Thousands of people completed a telephone survey from which we recruited roughly half of the adults to participate in the clinic cross-section, and some of them chose to bring their children, and they also participated. And then you can see in the follow-up years these numbers and these numbers. So I'll tell you why this is so small in just a second. So these are the repeated cross-sections, and this group is the one that came in after the first year and the second year. On the national sample, which is somewhat later, you can see the numbers get larger. Almost 6,000 people completed a telephone survey. Almost 1,500 people were followed longitudinally, and you got this very big number of the first cross-section, and then much smaller in the subsequent ones. So these are large studies, complex design, very, very big investment in money and time and organizational infrastructure. So the content of, for the first aim, the things we measured were diet-related psychosocial constructs. Those are things like, how important do you think diet is to health? Do you think it's important to follow a low-fat diet? Do you think about nutrition when you select foods? Then we got usual consumption of snacks, fruits, and vegetables over the past month. We did this using a food frequency type approach. We would say, over the past month, how often did you eat such and such? And then we asked them about their specific foods they ate in the previous day to try to get at whether or not they were eating Olestra chips, or in this case, any kind of chip, at the same time they were eating high cholesterol food. Excuse me, a high carotenoid food. And it's worth talking about methods here because measuring diet in free living humans is very difficult. And in fact, since I've done it most of my professional life, I can tell you it's horrible. And this study was designed maybe 15 years ago, 12 years ago. Some of the methods that we used were new because we had to develop ones. And some of the methods that we used were the so-called accepted standard measures, and we now know they're absolutely bogus. So one thing we'll come back to is the difficulty if you're monitoring food consumption in a population, how on earth you measure it. But for usual food in the past month, we had this standard five-item questionnaire, fruits and vegetables, which has been used throughout the US for many years. And we had to develop a snack questionnaire because no one had ever attempted to study snack food consumption in detail. So there are different types of snacks, and each type of snack might have a different formulation. Maybe it's regular fat, maybe it's reduced fat, maybe it's non-fat, maybe it's Olestra fat. So it gets very complicated in how you assess exactly how much of the white type people are eating. And we had to develop this method to measure co-consumption, which is a modification of a Sainer technique called a 24-hour dietary recall. So a 24-hour recall, you sit down with someone and you, in very great detail, go over exactly what they ate in the previous 24 hours. And if you just want to think about if you can do this in your mind right now, how easy it would be for you to go back to yesterday and report absolutely everything you ate in great detail, it's tough. And it takes a long time. It takes about half an hour. So we had to modify that. So we developed what we call the focused 24-hour recall, where we went meal by meal, and we only asked about the things we needed to know. So we only asked about snack foods, and we only asked about vegetables and fruit. And in the analysis, we wanted to make this population. We wanted a population inference. So every observation was weighted for both their sampling probability. This is when you make a phone call to a house. If there are 5 people living in the house, then everyone has a 20% chance of being sampled. So you adjust for the inverse of the sampling probability, and then you adjust those numbers to the age-sex distribution of the underlying community, so that the end results are actually inferences you can make about the entire community. And what we looked at were the age-sex specific distributions of of total snacks, and we looked at associations of demographic characteristics and the psychosocial constructs of snacks. And then we tried to get at this issue of co-consumption, and I'll just show you the results. So first of all, Olestra consumption was very low. The 15% of people reported eating Olestra more than once per month, and of the 15% who reported it, the mean number of servings is 3.6 a month, and if you look at the tails of that distribution, the 95th percentile was only 14 times a month. So it turns out that people were not eating 4 and 5 servings of Olestra per day. The 95th percentile, they're eating a serving every other day. So there was very low uptake, and you can compare that to other types of snacks, regular fat snacks and reduced fat snacks, where you can see most people report eating them, and they report them at much higher servings per month, 6 versus 1. And the people who chose to use these snacks were just about who you might guess would choose to use them. There were absolutely no surprises. Women tended to use them more than men. They were more likely to be used in sort of young adults, which, by the way, is where you see the highest consumption of these kinds of foods. They were much more likely to be used by whites and blacks, and their use increased with education. So this is exactly what you might think you'd see. They tended to be used more. They tended to be used more by people who were obese, by people who were not diabetics, and by people who had high serum cholesterol. So it all fits together. These are people who perceive themselves to be at some kind of nutritional risk, and they were using these foods for probably health reasons. And when you look at the psychosocial constructs, well, if people thought it was important to eat a low-fat diet, there was this huge effect. They were almost 4 times as likely to eat the chips. You see this very weak effect for the extent to which people think diet and disease is important. And this concept of nutritional salience is the extent to which people think about nutrition when they're actually eating food. And that was a very weak predictor. And co-consumption, I'll just walk you through how this works. So these are the percentage of people who ate foods at these times. And so most people ate dinner, and most people ate lunch, and most people ate breakfast, and then the snacking was less. And these are the percent of these people who ate a fruit or vegetable at that meal. So you see fruits and vegetables tend to peak at dinner and lunch and breakfast. Again, no surprise. High carotenoid fruits and vegetables really concentrate at lunch and dinner. Savory snacks, however, concentrate at the snack times, late night, mid-afternoon, mid-morning. And if you go over here, these are the percentages of people who reported eating a savory snack. This is pretzels or chips or anything like that with a high carotenoid food, and it's very low. And if you summarize all that information, it looks like this. So roughly 20% of the population reported eating a high carotenoid food with a snack chip at some point during the day. But if you look at the percentage of total carotenoids that are at risk, meaning the percent of all the carotenoids you eat that were eaten at the same time they eat a savory snack, it's a very low number. So not even 10% eating regular fat, 5% reduced fat, and almost nobody reported eating an Olestra snack at the same time they ate a high carotenoid food. So this issue about co-consumption turned out to be not much of a problem. There wasn't much co-consumption. So that was aim 1. So we got some idea who used it, and we got some idea of how much they used it and the eating pattern. So now we looked at the serum micronutrients in the population. So there was the clinic visit was 1.5 hours, and it was a fasting blood draw. So right there you can see there's an enormous participant burden. And at that visit, we collected information on diet, supplement use, sun exposure, BMI, smoking, all sorts of factors that we knew or hypothesized were going to be related to serum micronutrient concentrations. And the analysis was, again, we used a weighting approach. Everyone was weighted for the sampling probability and the age-sex distribution of the population. And the approach we used here is we fit multivariate models on the pre-Olestra time period, predicting serum concentrations of each of the micronutrients and carotenoids we were interested in. And I should say that using all the information we had, using dietary intake, supplement intake, age, sex, race, physical activity, and a number of factors that relate to serum micronutrient concentrations, we can explain between about 4 and 18% of the variance. So just doing the best job you can, you really can't explain very much of the variance in serum micronutrient concentrations. But we use those statistical models pre-Olestra, we fit them post-Olestra, and we added Olestra consumption to the model. So this allows us to look at the extent to which Olestra could increase the amount of variability we could explain in serum micronutrient concentration. And the results look something like this. Olestra wasn't associated with serum micronutrients in this sample. Absolutely nothing with carotenoids, nothing with lycopene. The only thing that was at all interesting was the slight increase in alpha tocopherol among the people who were so-called high Olestra consumers. And I want to point out the distribution of Olestra consumption per day. This is about the best we could do. The highest group was 2 grams or more a day and only 26 people. So we didn't have a lot of people who were high users from whom we could make a lot of inference. So there's nothing going on in micronutrients. And then we turn to aim 3, which is looking at change. So this is a different type of statistical model where you condition post-Olestra consumption serum micronutrients based on pre-Olestra consumption serum micronutrients. It's a much more powerful test as to whether an exposure is related to an outcome. And here's what this looks like. So you couldn't see any consistent pattern in serum micronutrients. A 10% drop in the highest group in carotenoids, not statistically significant, I should note. Random variability for the others, and this interesting increase in vitamin K. Olestra was supplemented with vitamin E and vitamin K and vitamin D. And this increase in vitamin K is probably just the fact that the foods were actually fortified with vitamin K, so that's what we're looking at. We also looked at the utility, as it were, of these foods. Are these foods doing what they think we'd like them to do to the population? So we looked at whether using them was associated with weight loss or serum lipids. And they were associated with about a 0.5 kilogram reduction in weight, a reduction in cholesterol and LDL cholesterol. This is exactly what we wanted to see. These two are statistically significant. This is borderline. And so it looked on the outset, based on the this sentinel site, that Olestra was not affecting serum micronutrients, and Olestra was having the efficacy that we wanted it to have in terms of nutritional status in the population. So in 1999, FDA looked at these data and they said, fine, you can continue marketing it, but here's what happened. Olestra sales were horrible. So they started high when they were released, and then, it didn't take very long until they tanked. My own feeling is because of the way they tasted and the way they were marketed, but perhaps P&G will tell you otherwise. And here's what happened. So P&G looked at those sales data and they said, we're done. Cease and desist. And so they abruptly stopped the study. They closed. So the Sentinel site was closed at the end of year 2, so we had 2 years of follow-up data. The national sites closed midway through the 2nd year. All the funding ended, and it's 2000. Really, nobody cared anymore. People were eating the food, so nobody cared about its outcome. And we never got around to publishing this stuff until 2006, when finally I got a postdoc interested in working it up. So, and these results are fascinating because they're exactly the opposite of what we got from the Sentinel site. So, first of all, there was evidence of reduced carotenoids. There was evidence of reduced carotenoids total, reduced lycopene, no effect on D or tocopherol, and reduced vitamin K were the last one we saw, increased vitamin K. And these are all, these three are all statistically significant. But also, we'll talk about this in a second, but the real question is, so there's a slight decrease in this high consumption group. Is that decrease biologically meaningful? And a 10% reduction in serum carotenoids, probably not. We also looked at the weight loss in serum lipids, and we saw, again, the opposite thing. We saw, in fact, it was associated with weight gain. We did still see the cholesterol effects, but they were much diminished. So that's kind of the story. That's what we did, and that's what we ended up with. I want to point out how expensive this was to do. So they stopped the study midway through, and we still spent over $27 million. And that's a breathtaking amount of money when to think about it, how little we got for it. So I will just generalize later to what that might mean. And there were a number of scientific challenges that we had to deal with in doing this post-marketing surveillance, and they're worth chatting about for a minute. So the designs are very complicated, and the protocols are hard to follow. And this is, I think, going to be true of any post-marketing surveillance activity when you need to measure intake and outcomes in individuals. And I don't see any other way to do it. Blinding was important. We called this thing the National Study of Nutrition and Health. And we said it was sponsored by a major food manufacturer, and that's all the information participants got. They had no idea it was a study of Olestra. And that was critical because we didn't want them to bias their reporting, the information we're getting from them, by the fact that they knew we were interested in Olestra. It turns out that there was very low exposure, and so in the end we had very few people who ate a lot of the chips on whom we could make inference. It cost a fortune, you saw that. We had a lot of measurement problems. Indeed, measuring micronutrient and carotenoid intakes from diet is extremely hard. We had to figure out how to measure snack foods and co-consumption. And the last challenge we had is because the study ended early, we had to do an analysis based on incomplete study data. So whether results we saw with the incomplete data would have matched the entire sample is unclear. And what are the lessons for Olestra? Well, I think there are a few. I think the most important is that the design of any post-marketing activity has to be hypothesis-driven. And you have to know what you're looking for to be able to go find it. And earlier someone brought up this question about statistical testing. What's critical here is that you have to have a hypothesis because that's the only thing that really the statistical test for which it obtains. If you have a hypothesis, you can test it with a known amount of power and you can give it a probability. If you go on a fishing expedition and you haven't articulated what your outcome is, then the probability of finding that outcome is relatively uncertain. It's hard to specify. There's a lot of ways to do it. You can sort of adjust the probability, but none of them are satisfactory. None of them are really as convincing as having an a priori hypothesis which you then go out and test. Exposure assessment of food is notoriously difficult. It hasn't gotten any. The techniques that we used 15 years ago, we know now are bogus, frankly, and the techniques that we need to use are extremely expensive and very burdensome on study participants. You have to have outcomes that are defined, feasible, and unbiased. So our outcomes were serum micronutrients, and I think they follow that criteria, but any outcome you're going to use will hopefully be one that's measured by a biomarker and not one that's measured based on some perceived response. And it has to be adequately powered. And here, powered to detect a biologically meaningful result. Now, we pulled 10% reduction in serum carotenoids, frankly, out of the hat. There was no basis upon which to say a certain percentage reduction in serum carotenoids would affect health. What I would now tell you is that a 50% reduction in serum carotenoids would probably have no impact on anybody's health. But this is This was a number we just had to pick, and we did. Then there are questions of funding. So we were funded by industry, and I can tell you that at the data analysis phase, as independent as I thought we were, and as we specified in our contract, I was actually trying to get a result to look the way P&G might want it to look. And I had to catch myself doing that. There is no matter how you look at it, if you are funded by industry, you're going to be influenced by that. And I think there's no way around it. There's no way to be fully objective. So whether you're funded by industry, government, or some independent foundation, I think is really important. And I would have preferred, in some ways, that we were funded by the government or by an independent foundation, and we had a different way to assure that we were insulated from anyone influencing our interpretation, be it either the community, consumer groups, or by industry. Because this all feeds into the regulatory pipeline, we discovered an enormous amount of problems in satisfying the FDA. The first is that the way you go about doing epidemiologic studies doesn't really follow the way you do randomized clinical trials. And the FDA was not set up for this. We make decisions about data for things like data exclusion analysis, which are not standardized. You have to look at the data and decide what makes sense, and then you make a decision to go with it. I'll give you an example. If we're doing an analysis and we find 1 or 2 observations out of, let's say, 5,000 that are having undue statistical influence, in other words, 1 or 2 observations which are so skewed that they change the result, then we remove them because we don't want to have an outlier having that kind of effect on our inference. Now, in a clinical trial, this doesn't go, right? You need to think about doing that. The result is subject to judgment. So whether there's dose response or not, whether there's a U-shaped response or an inverted U-shaped response, or whether there's a threshold effect, this is all in the eye of the beholder. The data documentation verification process for regulatory studies is enormous. And I think it can't work in this kind of study. There's no way to essentially verify the way you do in a clinical trial that the data you collected was from the person you wanted to collect it, and the data were handled with the same level of scrutiny and absolutely every step of the way that you see in a clinical trial. It's just unfeasible. And lastly, FDA said they wanted our data, and they wanted to replicate our analyses, which was ridiculous. But we gave them our data, and we gave them actually code that we used for analysis, and they didn't know what to do with it. They didn't have a clue. So the regulatory agencies are not set up for this activity. I think another issue is how are our research findings distorted? Because we had these relatively good scientific results. They were easy enough to interpret. But at the FDA hearing, I'll give you an example, anecdote was used. So Center for Science and the Public Interest, who decided that these chips were bad for you, got people in who would say, I took 1 bite of the chip and I had diarrhea for 3 days and I was violently sick. And honestly, that's FDA testimony. So anecdote is hard to deal with in this construct. Attribution bias, which I talked about earlier, if you look at the number of adverse reports from the telephone-based call-in that P&G had, basically it was a sort of flat line, except for these points when there were big media events, and then you could just see it peak, and then it goes down back to a very, very low level again. So if people think about attributing a common effect like diarrhea to something they ate the day before, they'll say, oh, that's why I got it, and they'll phone in. So that's just attribution, but it's not really real. Then there's ideology. It doesn't matter what science you throw at people, natural and organic is the way to go. I don't know how this plays out in Europe, but I can tell you in the US, there's all this ideology about food, which has very little scientific basis, but it's powerful. And lastly, media. The media is interested in reporting sensational results and not really interested in reporting balanced, thoughtful results from scientific work. So the combination of all of these things can really distort research findings, and we found that. And lastly, there are some limitations to observational epidemiology, and they're real, and you can't get away from it. One is reproducibility. So we spent $27 million doing this study. Are we going to do it again for another $27 million to show that it's reproducible? That's tough. But if you look at the breadth of epidemiologic studies looking at any particular sets of questions, there's a lot of variability. And it's only after you've done many studies and think about them, interpret them, that you think you've got something reproducible that you want to make an inference about or want to make a public health recommendation from. Generalizability is a problem because the people who choose to participate in your study, no matter how much money you pay them, are not generalizable to the usual population. Everything we do incorporates measurement error, and by the way, everything laboratory scientists do incorporate measurement error too. They just don't like to think about it. But there's enough measurement error that sometimes you can't see a result even if it's there, using the best techniques you have for measurement. And lastly, bias and confounding can work themselves into an epidemiologic study in all kinds of awful ways. And we do everything we can to try to avoid it. We think about what the biases might be. We think about how to recruit people without bias. We think about how to measure without bias. And we think how to analyze without bias. But we never know. So I'll stop there. But that just gives you, I think, a sense of the challenges of doing post-marketing surveillance on something as simple, really, as Olestra. And when you think about how you might do post-marketing surveillance of something like genetically modified foods, I think it's, to me anyway, it looks enormously challenging, and I don't even know if it's feasible. And I guess we'll talk about that later.
Thanks, Alan. I was just thinking as you were going through your talk, anytime you want to send me a registered letter, have we some questions there, please, from the audience? Well, I have one, and it would strike me as being probably the most obvious question. You went through a list of things with regard to what you would need for a post-market monitoring scheme. The first thing you said, you'd need a hypothesis, and the scheme should be hypothesis-driven. Surely, if we have a hypothesis, that should be looked at pre-marketing. I don't know if you want to comment on that, or maybe
the hypothesis comes from pre-marketing.
Sure.
The pre-marketing can never look at the impact of releasing a food in the population. They can look at the impact in a randomized trial on a small number of people. They can look at impact in animal models. They could do statistical modeling. But they could never really incorporate what happens in the real world when something's released. And so that I think is where the hypotheses need to come from.
Okay. Michelle?
Do you think that we can afford to do this for new foods?
It's fantastically expensive. Let me think about that for 1 second. I think I can imagine setting up a kind of surveillance system, and I mean active surveillance, like incorporating people into research. If there were multiple novel foods introduced into the market, you would have an infrastructure set up to be able to evaluate them. I think on a case-by-case basis, instituting something like this is phenomenally expensive. But if there were 10 or 15 novel foods being introduced at any one time, you could have the same approach. And I don't think you'd have to modify very much of either your exposure or your endpoint assessments, in terms of cost anyway, that you couldn't do them all at once. So it's sort of a thought.
Sandy, do you think that this is what the EC are thinking? That this, you mentioned it as a case-by-case you know, post-market surveillance. But is this a structure? And if so, how are we going to afford to do this?
I think some questions can be answered more cheaply than others. If you're going to ask about going to whole populations, looking for the small proportion of people who are exposed to a particular product, and then doing this kind of analysis on them, then yes, it's going to be expensive. If you want to get a handle on exposure, just simply to confirm that exposure levels are within a particular limit, without bothering about the end effects and all the clinical data that we've collected here, then I guess the cost would fall quite substantially. So I think there's no single answer to the question, because the need for post-market monitoring and the nature of that monitoring would be case by case, judged case by case. So I think the Olestra case tells us that if you are going to have this kind of question, then really it's going to be very difficult. But as I say, as I tried to say yesterday, there are different types of levels of question that you might want to answer through post-market monitoring. Some of those will fall out more easily than others.
Mm-hmm. And I just want to throw out that one of the things I hear from people because I talk about food, people talk back to me, because of course everyone eats and everyone knows about food, is the ideation people have about such and such food is going to increase my risk of cancer, or such and such food is going to increase my risk of some horrible endpoint disease. And these are unanswerable questions. So people really were concerned when Olestra reduced. People were actually talking about, oh, it's going to increase my risk of cancer, I'm not going to eat that. I mean, forget mechanisms or what have you. Evaluating that kind of question is basically impossible. And so post-market surveillance is really limited, I think, in its ability to look at outcomes only up to a certain point. And they have to be relatively acute outcomes, or they have to be intermediate outcomes that we think have some inference to disease at a later point.
Okay. Just one other comment. I'm just going through some of the comments you had with regard to post-market monitoring. But exposure assessment, you said, of food is notoriously difficult, right?
Food is notoriously getting people to report what they eat is notoriously difficult.
Right.
Yes. Yeah.
And I'm just wondering, and someone in the audience may wish to comment on this, how much easier is it going to be with regard to animal feed?
I hope you're doing it. Yeah, just from the animal feed perspective, it's a question we've had the whole time, because we are supposed to be doing post-market monitoring. But I mean, we've already made it very clear that it's an impossible job for us. It's just not something we get involved with. So I'm also very interested, in the discussions between the biotech producers and the shippers and that. But the feed industry in Europe, it just is unfeasible. We just couldn't do it. So from the point
interview a random sample of pigs. I mean, it's
yeah.
Right.
Is there anyone else who would like to comment? Sorry, Marie.
With all the traceability that's going on in Europe anyway, in the food chain in general, I don't know. Is it really that impossible? Feed producers for salmon feeds anyway, from what I understand, are sort of keeping track of batches that they're using and things like that.
Well, is the ultimate goal to monitor the health of the animal, or to monitor the health of humans?
No, that's feeds for animals. That's Norway might be a
No, but I mean, so what the animals are fed, let's say, but then you have to figure out what humans are eating, which fish.
Yeah, no, I was thinking of animals then as sort of a surveillance tool.
Animal health.
Animal health.
Animal health.
Yeah.
Was that your question?
I mean, they're eating the same feeds day after day, so exposure might be more constant than it is with humans.
But I'm not an animal guy, but I don't know what outcomes you might use to have any inference to humans.
They may not actually be eating the same foods day after day because, well, particularly in the pig scene and poultry scene, they're least cost diets, so it'll be based on the ingredients that are least cost at a particular time point. So there are difficulties there. Yeah.
Norway is a bit different.
Question here, Rick.
Yeah, and that was an excellent lecture and a lesson for me because I have never heard a presentation specifically on PMM on food before, so thank you. When I think about it, and I showed a picture at the end of my talk, which is here are the products that soy is in, and that's a very small sample. And then you think, for instance, if you wanted to monitor health effects of Roundup Ready soybean or somebody's other enzyme-driven soybean, or you wanted to measure something about maize, because maize also goes into many different foods, and we really don't have a good way to monitor even what's the percentage of maize or soybean in a huge variety of foods that are out there. So to me, just from a practical perspective, it's an undertaking that 27 million will be paled. You know, it will be well over $200 million, I think, if you want to do it accurately. But I possibly am way off. So it's good to have a couple of people who know something real about post-market monitoring here. And I think after the break we'll have
I just want to say the question there is if you have a biological assay that tells you something about individual level exposure, then you're in good shape. But if you don't have that, then I think it's unfeasible.
Just another one of your comments. So
I'm full of comments.
It summarized everything with regard to post-market monitoring for the GMs. But you said it should be a well-defined, unbiased and feasible outcomes, actually. And you mentioned with regard to bias, or maybe unintentioned bias, because it'll be a study performed by the company in question. Is that likely to be a problem with GM if there is a post-market monitoring system, if it's not carried out by an independent body?
I think it's a problem that people even know what you're looking for.
Yeah.
So people in our city didn't know it was about Olestra, and we thought that was critical. I don't know how you could even mount a study to look for effects of GM foods without people knowing what it was you were after.
Yeah.
And so then the role of attribution comes in, and the role of bias, which is participation, also comes in. So, you know, all of these things, I think, have solutions based upon the specificity of your outcome and exactly the question you want to ask. So the reason I started with the most important thing being a hypothesis-driven exercise is at least then you can design the study that answers a particular hypothesis. It may be that the study can't be done. You know, that happens too. You come up with a study with a sample size that's 5 million people, and you say, well, no, this isn't going to happen, for example.
Sure.
Yeah.
Sure. Yes? Question down here.
Hi, I'm Constable Vanessa. I just have a comment with regards to responsibilities for doing PMs on GMs. If we're going to look at the consumption of food products containing GM ingredients, then you've got to have the interaction of the food industry who are putting the actual products onto the market. Then you'll have to have the cooperation of the manufacturers who are actually producing the GMs. And so I find it very difficult to understand who is going to take responsibility to do any of this marketing anyway. And if such an activity is undertaken, there's going to have to be governmental, independent overseeing, actually, of it, because I find it completely complicated to do. Just to say, coming from the food industry, but you have different ingredients from different suppliers in your foods, and I find it very complicated.
Okay. One question, there's a question down the back.
Yeah, and a question. So, looking back retrospectively on what you did and the money that was spent on this, do you think the resources that were put into this to do the monitoring were proportionate to the risk that was initially found, or
Well, I think there are two relative ways to think about the money. I think the way Procter & Gamble was thinking about it was they'd already spent somewhere around $800 million developing this product. And the FDA said if you want to keep it on the market, you have to do this. But they were not very happy about the amount of money, I will tell you that. when you think about the risks of Olestra, I guess my bias going into it is that they were going to be minimal. And my interpretation coming out of it many years later is that they really didn't exist. There simply were no risks. There really is no biologically supportable inference related to serum lycopene being reduced by 20%. It simply doesn't matter. And so, no, I mean, it's a huge amount of money to try to show something is safe but everything you knew about it going into it would suggest it's safe. And I think GM Foods has a bigger problem in that, at least from what I heard today, I think that last talk was fascinating. I've never seen so many no results in my life. No effect, no effect, no effect, no effect in all these different models. Then what you're going to look for, monitoring. I mean, I don't know what you're going to look for when you monitor GM Foods if it doesn't do anything.
Just, unless there's another question there, if someone wants to be thinking of one there, just one with regard to power. Before we start any experiment, you need to know that you have sufficient power to determine the difference you want to determine, right? In that respect, would it be better if post-market monitoring was to be done in a situation, should the product only be released into a test market in the first place so that your sample size doesn't need to be as big?
No, your sample size, the inference is based on the sample size, not the amount of people you're doing it in.
Sure.
So if you want to show a 10% reduction in serum carotenoids, for example, we calculated the number of people who had to be in the study, and that was based on the total number of people, the total number of times we were going to do the measurement. It doesn't matter about the market. I think the critical piece in all this is what's the outcome.
Sure. Okay. We have a question there, Sandy.
Not so much a question as a comment, just a reminder that from yesterday we were looking at what the Commission has put down on paper and saying these are the conditions in which we think there might possibly be a justification for post-market monitoring. And those three cases were to confirm that specific conditions of use are being adhered to, where they've been defined for a product that's being authorised, which is nothing like what we're talking about here. To confirm the predicted consumption of the food or feed, which was kind of like the 5-sterol example I described yesterday. And then only the third one takes us into the Olestra territory, which is relevance and intensity of effects that have been picked up during the pre-market assessment. So this is kind of the nuclear option, if you like.
It is the nuclear option, yeah.
There might be, but there are other types which would be certainly easier and cheaper and more straightforward to address. The fact that 3 individual member states have carried out monitoring for pharmaceuticals as a novel food tells you this is not something that's hugely expensive and requires large amounts of private money to make it work.
No, you're right. Monitoring the use of these things, as long as you know how to measure them, is relatively straightforward. It can be done inexpensively. It's as soon as you add outcomes that it gets complicated. And I must say, even if you're trying to confirm the positive outcome, the effectiveness of the food, most of these foods have some sort of, well, I take that back now. GM foods have no health claims. And usually you're thinking about food that has a health claim. So GM foods don't even have that. So there's no effectiveness to measure that I can know of. So as long as you're not looking for, as long as you're just trying to understand who uses it and how much they use it and what patterns they use it, that's inexpensive.
We have another question here.
I'm just going to disagree. I'm going to be leading a project where we will do an effectiveness study.
On GM foods? I mean, in humans? Like
on golden rice, yes.
Oh, golden rice. Oh, well, yes, of course. Well, that has a that has a health claim. Yeah.
Sorry, but even before we just finished that. Even before golden rice, in the projects in Africa, we've already done effectiveness studies on orange sweet potato. Again, that's a simple thing that you're measuring a single item.
I was thinking about GM foods, meaning you genetically modify the soybean in one gene to produce a protein. I don't think there are no inferences you can make and benefits to the humans consuming that particular soybean versus another soybean. things that are genetically modified to produce micronutrients, for example, is a totally different story. Yeah.
Okay, we have one last question here.
I think the Olestra case is very impressive in terms of efforts, costs, and the outcome. I wonder if you are aware of any other recent examples of post-marketing monitoring with food. Do they provide a similar picture, or are we left with this one example?
I don't know of any studies that have gone to the sort of the Cadillac. What did you call that? Not the Cadillac model, but the nuclear option. I don't know of any studies that have gone to the nuclear option. So I don't think there are. I'll use my Mac. Yes, I'm sorry. I don't think there are any other models. Is anyone aware of anyone else?
Did Simplest have to go through something like this? Was it on the market long enough?
It did, but I don't remember if they actually looked at health outcomes or if they just looked at the. I think what they did with that is they showed that the use was roughly 1/10 of the use that they felt was potentially harmful, and so there was no need to do it.
Just in my talk later, I'm going to mention aspartame as a sort of BMN. Then I'm going to mention Olestra, but luckily I don't really have to go into details because you've done it much better than I would have done. And I'm going to mention the phytostellar Olestras, which is a novel food in Europe. And another one.
Did they use the nuclear option, or were they somewhat more restrained?
No, yours is the
That's an advertisement for the next session. Okay, thank you, Dr. Kristal.